Hello everyone, and welcome to today's on-demand webinar, Axiom Analysis Suite for Genotyping Analysis of Axiom Microarrays. My name is Michelle DeRozier, and I am the Field Bioinformatics Specialist for the North America FAS team. We're going to cover an overview of the Axiom Analysis Suite software, what it is, why you want it, and where to find it. Then we're going to jump into a demo where we're going to cover the basics of getting started and running an analysis, along with the features and capabilities. Axiom Analysis Suite is a free software that you can download directly from our website. Axiom Analysis Suite has the capability to run not only genotyping, but copy number as well. And although this demo will focus solely on the genotyping aspect, there is another webinar that will focus on the copy number function. Axiom Analysis Suite is not only easy to use, but it has superior graphical interface and multiple exporting options for almost any need. Axiom Analysis Suite is also flexible. It isn't just specific for human. It can run plants and animals as well. And as I mentioned, it's easy to download from the website link below. As with any software, Axiom Analysis Suite has some minimum recommended settings. Generally, we support Windows 7 and Windows 10 with a minimum of 32 gigabytes of RAM. Now let's jump to the demo. Once you have downloaded and installed the Axiom Analysis Suite software, you can open it directly from the Start menu under the Thermo Fisher folder, or if you created a shortcut on your desktop from there. When you open the software initially, you'll be prompted to pick a profile name. If this is the first time that you have opened the software, you will be required to create a profile. Once you pick a profile, it'll bring you to the home page, which is to where you would run a new analysis. The first tab that you're going to want to visit is this Preferences tab. This is where you will download all the library and annotation files for analysis. Library folders are defaulted to the location shown here. However, this is user-defined and can be modified or changed. The Update button here is where you're going to go to choose and download files that are needed for the array of your choice. One thing to note is that this does not apply to custom designed arrays. Those files need to be manually installed. Over time, Thermo Fisher will update library files for modifications. There is an option where you can say to check for updates at the startup. If this is checkmarked when you load Axiom Analysis Suite, it will prompt a reminder that library files have been changed or updated, and you need to go into this NetEffects library file location and update those files. There are other preferences on this tab that we're not going to cover, but you can please reference the manual to know more. When you are ready to download and install a library file, you can simply click the Update button here, and it will bring you to a login page for NetEffects. This is a separate login and user account that needs to be created specifically on our NetEffects website, which you can find on the general Thermo Fisher page. Once you log in, you'll be presented with a list of our commercially available arrays for download. As you can see here, this is a list of complete Axiom arrays that are available to download within Axiom Analysis Suite. Those that have a green check are already installed in the library folder. To install a library file, simply click on one and press OK. This middle tab here is called your Dashboard tab. This is where you can access your previous analyses that were run. You can also browse to older analyses by using this option at the bottom here. This is for analyses that maybe you have moved to an external drive or a server location. We're going to move to the main tab called New Analysis. This is where you will load and run data for genotyping. The first step in running an analysis is to load the raw data or cell files. This can be done by clicking this first folder option here that says Import Cell Files. If you have both cell and sample information called ARR files, you can load that information together from these two options to the right. Although it's not covered in this webinar, you can reference the user manual for more information. If you load cell files and the library files have already been downloaded and installed, the array name will populate automatically up here. If you try to load cell files where the library and annotation files are not loaded already, you will get a pop-up window that says that your data cannot be loaded because there are missing files. In that case, you will need to go back to your Preferences tab and link out to the NetEffects site and download the appropriate library files. Once the data is loaded, you will choose a workflow analysis to run. Depending on the array, you may have multiple workflow options to choose from. However, in this webinar, we are going to focus on the best practices workflow, genotyping. The best practices workflow is an automated, stepwise method of genotyping that incorporates multiple QC metrics to make sure users get the most accurate and confident genotyping data output. Here is a screenshot of the breakdown of the best practices workflow. It's an 8-step program, and steps 2 through 8 are automatically executed when using the Axiom Analysis Suite software. Essentially, the user is responsible for grouping samples into batches for appropriate genotyping. If you are interested in learning any more details about each step individually, please reference the user manual. Once you load your data and choose the best practices workflow option, you will see a myriad of analysis and threshold configurations that populate on your screen. Some of these options will only apply to certain species. The inbred penalty, for example, will only be used for certain ag bio species. Although we are not going to go through all of the options here, and we will be using the defaults that automatically populate, you can find more specific information in our user manual. Once the workflow setup is completed, you will choose a folder to save your data and a name for the specific analysis. Then you will click Run. Depending on the sample size, the workflow type, and the computer space, the analysis time will vary from situation to situation. Once you click Run, your screen will then flash to the Dashboard tab, where it will have your batch name and a progress bar that tells you the status to completion. Once your analysis is completed, you can then open it by the Open Action button on the right-hand side. So we're going to open up the batch that I had run previously. Once your batch opens, you can view all the details about the analysis that was run. To the left here, you will see the descriptive tabs, starting with Summary. The Summary tab will provide you a high-level overview of the batch details. It will provide you basic information like the batch name, the array name, the workflow type used. It will also provide a breakout of the sample summary, so the number of samples that passed each of the QC cutoffs that are used in our best practices workflow, and an overall pass rate. It will also determine gender of the samples, and then put all of that information in a table here labeled Plate QC Summary. It will also show batch details related to probe set and SNP or marker performance in the tables here. An important note is that Axiom Analysis Suite will break down the markers or SNPs into cluster categories. There are 6 types of SNP clusters, and depending on the array species type, those categories are broken into recommended and not recommended. Here is a slide to visualize these categories more clearly. The top row are what we consider good cluster types and recommended, and the bottom row are poorer cluster types and not necessarily recommended. As we go through the webinar, I will highlight some more examples of these. But if you're interested in the details, please reference the user manual or the Axiom Analysis Data Guide. The next table we're going to look at is the Sample Table tab. The Sample Table tab is where you'll see a list of each individual sample run in the batch and its individual performance. It will list out the QC metrics, the gender, the plate barcode, as well as position. The Sample Table tab also has additional functionality. It will allow you to create metric plots, which can be found under the QC Analysis drop-down menu. You can make scatter plots, box plots, or plate views. When you choose the type of plot to make, it will be presented with a list of metrics to choose from. You just simply choose which one you want for the x-axis and the y-axis and press OK, and that box plot or that scatter plot will show up on the visualization side at the end. The Sample Table tab also will allow you to add in additional columns of information by the Show Hide Columns dropdown. If you click that, you'll see additional information that is not necessarily added to the table that it exists as a default. Anything that's checkmarked is something that's already loaded. If you were interested in seeing the number of samples that passed as a column, simply add it and it will be appended to the end, right here. You can also export these tables for tracking purposes if desired. You can do a current table if you were filtering all data, or if you wanted to highlight specific samples by doing Control, click on the samples of interest, and then click Selected Samples Only for export. There are multiple ways to filter the data in the Sample Table tab. You can either double-click on individual columns and sort that way. So if I double-clicked on the Pass/Fail column, it will filter so all the failed samples are at the top. I can filter by QC computed gender, in which all the females will be sorted first, or the opposite way around. Another way to filter is to do the Filter drop-down menu. If you choose the Filter drop-down menu and Manage Filters, it will allow you to pick a column to filter by. So if I wanted to do a filter by dish QC call rate and do only C samples that were above 0.90, you will then see that only the samples that are loaded meet that metric that I just filtered. You can then also clear filters by right-clicking on a column and do Clear Current Column Filter. There's other functionalities in the Sample Table tab that we won't be able to go into today, but again, please reference the user manual for additional information. We're now going to move to the Probe Set Summary Table tab. The Probe Set Summary Table tab breaks the analysis down to the probe set and SNP or marker level. This table provides an individual call rate. It provides a conversion or cluster type. It also provides whether this cluster conversion type is part of our best and recommended category. There is additional functionality in the Probe Set Summary Table tab. Much like the Sample Table tab, you can add extra information based off of the Show Hide Columns dropdown. By simply checking on an option here, it will load that column of information and append it to the end of this table for visualization. The Probe Set Summary Table tab also has the ability to filter, like the Sample Table tab. You can choose filters from the Filters dropdown by managing filters here and choosing the columns of information that you want to filter by. Or you can right-click on the column that you're interested in filtering as a shortcut. So if I only wanted to see the clusters that were associated with the best and recommended, I would right-click on that column, choose Filter, and say that I want to see only data that has a 1 in it, which means not only is it the best probe set for that marker, but it also is part of a recommended cluster category. If I click OK, you'll see that the data will go from a little over 900,000 probe sets down to 881,000 probe sets. And you'll see that this little funnel, which indicates that a filter has been applied, and only the data that has a number 1 is associated in this table. If you were interested in exporting the data as it was, you can certainly click Export this current table, and it will only export the data with that filter applied. In the Probe Set Summary tab, you can also create probe set lists and filter lists by probe sets, as well as export genotyping data, but we're going to get to that a little bit later. Let's move to the right side of the screen, which is the Data Visualization section. Axiom Analysis Suite will provide several auto-defaulted visualization plots for any batch run. The first two are basic box plots of the performance based off of barcode for the 2 QC metrics that are in the best practices workflow, Dish QC and QC call rate. This will provide you an immediate visualization as to whether or not there is a specific plate that is functioning different from the rest in the batch. The next tab is what we call our QC call rate versus our Dish QC. This shows sample-based performance across the Dish QC and QC call rate results. You can adjust colors and shapes, but with different identifiers. For example, if I wanted to color by barcode, I would simply click on the drop-down menu and pick barcode. And if I wanted to shape by whether that sample passed or failed, then I would just find the pass-fail option. This gives an indicator as to whether or not there's a grouping of samples or a plate that maybe isn't performing as well as the others in the batch. The next 2 tabs are plate views. The first one is the plate view of the Dish QC performance for all the samples run. As you can see, anything in red does not pass the default metrics for that QC performance metric. The same with the plate view for the QCCR. One thing to note, that if a sample doesn't pass the Dish QC metric, it will not move forward for a QC call rate metric. That is why you see these empty blocks here. These are great tools to determine if there's any sort of plate effects from automation that are occurring. You can get great trend information as well. The next tab we're going to look at is the Cluster Plot tab. If you go back over to the left-hand side under Probe Set Summary Table and highlight a probe set of interest, The Cluster Plot tab on the right-hand side will dynamically change to show each specific cluster plot for every probe set that you highlight, where each triangle and circle within the cluster plot represents a sample that was run in this particular batch. If you're interested to see which samples are correlated with specific genotypes in a cluster plot, you can highlight and lasso them by right-clicking and dragging to make a circle around a specific set of samples. Those samples will be highlighted in pink, and then you can go over to your Sample Table tab on the left-hand side, and those samples highlighted and lassoed on the right are now blue in the Sample Table tab. The reverse process is also true. You can highlight multiple samples in your Sample Table tab and see them populated in pink on the right-hand side. If you were interested in sorting your conversion types, you can double-click on the conversion type column, and they will be organized by conversion type as well as the pre-existing filter we made of best and recommended. That way you can get a visualization and an idea of what the 3 conversion types for best and recommended look like. There's mono high res, no minor home, and poly high res. Axiom Analysis Suite also has the capability to export cluster graphs. If you're in Probe Set Summary Table and choose the Export dropdown, there is an option for cluster plots to a PDF file. You give it a file name. You can either choose all SNPs from the current table or a random set of SNPs. You can choose the amount of SNPs that you want to export, and you can either choose all conversion types, or if you're specifically interested in just an example set of poly high res or no minor home or mono high res, all of those are available options for exporting. Axiom Analysis Suite also provides the functionality to manually adjust calls. For example, in this particular cluster plot for this particular probe set, you see that there is a sample that is being no-called. If for some previous knowledge you are convinced that this should be either an AA, an AB, or a BB, you can right-click on that sample and you can manually change the call to what you think or know is the right call. Another very valuable function of Axiom Analysis Suite is creating and using SNP lists for filtering data. If a user is only interested in data from a specific set of probe sets, then it may be beneficial to only look at that data in Axiom Analysis Suite and not be overwhelmed by anything else. There are a couple of ways to do this. The first method is just generating a text file with a list of interested probe sets with the probe set _ID as the header. Once that file is created, you can then go into the Probe Set Summary table of Axiom Analysis Suite under Manage Probe Set List dropdown and choose Import Probe Set List to Batch. You then browse to the text file you just created, click on it, and open it up, and you'll see that your probe set count goes from 900,000 to whatever the filtered amount is. Click Yes to filtering with that probe set list, and you'll see that your count has now gone down to 881,000 probe sets based off of the probe set list that you just imported. Another option for creating a probe set list is directly from a batch that you have just used to analyze. If you know in the future you're going to be analyzing all the data associated with this customer, this lab, this group, based off of a specific set of probe sets from the data, you can create a probe set list from the data itself. This is done by filtering the data based off of a specific metric. For example, if I knew moving forward in the future, I would only want to look at data that was associated with the best and recommended probe sets, I would filter to only see the best and recommended probe sets, which we had discussed before. And my row count goes down to 881,000. Then I would go up to the Manage Probe Set drop-down option and say Create a Probe Set List from the table. If I choose that, I can simply give it a name. And press OK. That has now created a probe set list in the same format I showed before with the text file. And in the future, if I'm analyzing another set of data and want to use that specific probe set, I can load the data under Manage Probe Set List, input that probe set list, and the future data would be filtered off of that. As we move back to the visualization section of Axiom Analysis Suite, every tab up to the cluster plot tab is automatically generated when you run a batch analysis. As you can see in my example, I have extra types of visualization graphs that have been generated. As a reminder, this can all be done from the Sample Table tab under QC Analysis drop-down option. You can do scatter plots, box plots, and plate views. As you can see, there are a couple of other options under the QC Analysis dropdown, like concordance and batch allele consistency, that are not going to be covered here. But please reference the manual for more information. Exporting the genotyping data will be the final topic highlighted in this webinar. To export the genotyping data, you need to go to the Probe Set Summary Table tab and click the Export option drop-down list. There are several options for exporting data, and we are interested in the genotyping data export. Once chosen, you are provided a list of options. Depending on the downstream analysis pipeline, we offer several output formats such as text, VCF, and Plink. By default, the software will only export the best and recommended probe sets. However, if you have a specific probe set list that you want to export by, you simply browse and scroll to that probe set file and load it in. You can export only specific samples as well. If prior to choosing your export option in the Probe Set Summary Table tab, you go to your sample table and highlight only the specific samples you're interested in, then go to the Probe Set Summary Table tab, go to Export Genotyping Data, and then choose the Selected Samples Only option. The genotyping data for the samples that you highlighted will be exported only. As you can see, we also provide multiple call output formats: forward, call codes, and numeric call codes. You have the option to include the confidence and the signal for each probe set. You can choose the location for your output file and choose a specific name as well. You want to make sure that you have the correct annotation loaded for the array that you're using, and to choose an identifier. We recommend the probe set ID. Although there is default annotation information checked to be exported with your genotyping data, you have the flexibility as the user to choose as much or as little information that you want to be exported as well. Once you choose all the options that you want for your genotyping data export, you can simply click the OK button. Once the data export has been completed, you can scroll to the location where you saved your export file and open it up. And as you can see, you have your probe set IDs, the genotypes for every sample, and the extra information that you included as annotation options. This covers all the basic functionality in the Axiom Analysis Suite that I wanted to highlight in this webinar. As a summary, Axiom Analysis Suite is a highly flexible, user-friendly software for multi-species genotyping as well as copy number analysis. Axiom Analysis Suite incorporates multiple steps to make sure data are of the highest confidence and quality. Axiom Analysis Suite has additional advanced functions that were not covered during this webinar, but can be found in the user manual, such as inbred and polyploid settings for complex genomes, advanced analysis for off-target variants, the batch allele consistency test, and the concordance test. If there is any interest in obtaining the demo data used in this webinar, please feel free to reach out to our world-class app support team, affysupport@thermofisher.com.